A warm good morning to everyone. So I think I'm audible and clear and loud, and we will be taking a step forward from what we have learned in the previous class. So basically, now it is more of a concept where we had started with basic electromagnetic spectrum and the various properties of the electromagnetic spectrum, which we have capitalized depending on the wavelength specific, depending on the energy specific to it, and then coming to its wave nature of the electromagnetic spectrum, manipulating the wave nature to our benefit and get a structural informations from the sample or the molecule of your interest which you are looking to study. Now, taking a step forward to that, we are also interested to understand more about the structural orientation. See, in the previous in the previous one where we had the circular dichroism session, there it was more like there has to be an optically active compound or molecule in your sample. It may be a chiral carbon, and because of this chirality, what was happening, the rotation effect was happening, right? Now if this rotation effect is only one portion or one part of it, which was telling whether it is like a dextro or a levio rotatory, but that is not enough, or that information is not sufficient enough to give you the structural idea. So there is another you can say technique by which we are going to understand, and that is through X-ray crystallography. Now just a brief idea. I think you must have already had your classes of where the structures of protein, how complex a protein structure looks like with its various primary, secondary, tertiary and quaternary structures are there, right? And as the complexity of these structures keeps on increasing with the number of folds, the problem arises because they become like densely compact into a small you can say a spatial area, or rather to keep it like in a small space, more number of folds means more protein layers are there, correct? So the structural identification of these complex structures are not very easy, and that is why X-ray crystallography has a little bit more advantage compared to the previous CD, right? So crystallography is a technique which is used, basically if you say grossly, it is used to understand the structure of a protein. So why I'm talking specifically of a protein here, because proteins are you can have lot of structural orientations of proteins. You can have different types of proteins. Your proteins can bind with other ligands also, so making it a very complex structure to elucidate, and we will be taking it step by step forward. Right now, if I talk of like introducing you to the protein structures, I think the protein structures and the structural determinations, you have the primary, secondary structures, and then comes different sheets, alpha, helix and all, right? So these things are well versed with you. I presume that you already had this area covered in your class, and with that structural idea well and clear at the back of your mind, we are trying to find out how we can elucidate those structures with this today's technique, right? Now why these structures I'm emphasizing on? The basic reason is, I think all of you can very easily correlate that a protein, when it is denatured due to temperature, due to pH or any other chemical reagents, so what happens is basically the structure of the protein gets disoriented, right? Now when a protein's structure is broken, to be very simple, when you break the chains or the foldings of the protein, what happens, that protein becomes a linear or sequence of amino acid chain, that's it. But the problem biggest with this is the functionality of a protein is very much to its structure and its foldings. So meaning, if I make a protein linear, what happens is the function of that protein is lost, and that is why the structure identification, elucidation, or you can say determination of a protein makes it a very crucial point, right? Now, if like in a protein, as you can see here, there's one very complex one, and you will see a lot of foldings are there, and there will be a lot of angles are there, and these angles and protein foldings we need to understand to its basics. So we'll take it a gradual step to understanding how this protein folding can be deciphered. But just to give a broad idea, so what happens is when you use this crystallography technique, what happens is you use x-rays, and then these x-rays are passed on to your protein of interest, and what happens is you get a Ramachandran plot. So it is similar to this plot, and in this plot, as you can see, grossly the areas are divided. So you see a lot of segmented areas. You see 4 quadrants, and in this 4 quadrants, there's grossly, I know if I have signals in this region, my sample will have a beta sheet. If it is in this region, then a left-handed alpha helix or right-handed alpha helix. But this is not as simple as I'm saying it. And why? Because if you you'll all appreciate, because the proteins are not very simple structures. So what we have to do is we have to understand first the structural orientation, and how this structural orientation can be narrowed down to the atomic level, and from the atomic level what informations we can decipher by passing an x-rays onto this and getting an scattering basically upon passing through these structures. And once we get this scattering, what we get is we get spectra. So the spectras have certain things to tell us, and we have to understand what these spectras or these patterns of spectras are actually giving us information, right? So you have to always keep in mind the crystallographer who is doing this is not trying to determine the molecular composition, but they are trying to determine the structural orientation of a protein. So this is something which you always should keep in your mind, that it is not this technique is not in practice to determine the molecular composition, so what it is made up of, but it is more like what is the structural orientation of the protein of interest, right? Okay, and this is basically the plot which I was trying to show you. Now this is the electromagnetic spectrum, and in this electromagnetic spectrum you see depending on the wavelength, depending on the energy associated, there are different grossly areas which we discussed in a previous class. Here what we will be more interested on this region, x-rays, and x-rays, as you can see here, is the wavelength will be a little less or smaller compared to your visible spectrum. And when you talk of like visible spectrum, you have something around 400 to 700 nanometer, but x-rays will be, you can see here they are lesser than that. So that means they have more power, first of all, or the energy associated with this. Secondly, the x-rays will be, the wavelength of the x-rays will be suitable enough for this type of studies. How it is suitable, I'll come in a while. But you have to understand why specifically x-rays we are talking of, because we could have used other rays also, but x-rays are suitable in terms of identifying this. So this is basically the wavelength, and this is 1 lambda. So one you can say the starting and the ending in the same phase, and that annotates 1 wavelength or lambda. And in this, what we are trying to do is we are using this x-ray as a probe. Basically we are using as a tool which goes into your sample, and then it gives you some structural informations, right? Now again, coming to the point, why x-rays? So to be very specific, x-rays, if you see the lambda or the wavelength of an x-ray is very apt or in the range of the atomic distances. So if there are 2 atoms in a lattice, okay, so the difference or the distance between these 2 atoms, the wavelength of the x-ray fits into that range, and that is why x-ray is able to identify or give that information, because it is the wavelength is in that range. If you are using a wavelength higher than that, it will not be effective enough. Now it will, the X-rays will go to this lattice, or basically we are saying in a crystal, and crystal of the protein, and in this crystal what happens is that it will experience diffractions and different diffractions. I'll come to this, all of these points one by one. So basically it will experience diffractions, and once the diffractions are happening, depending on the patterns of diffractions, which again depends on the lattice structure, so you collect this diffracted X-ray onto a detector, and the spectra is drawn, and from the spectra you get two informations: what is the intensity of X-ray which you are experiencing, and what is the angle of diffractions which you are experiencing. So based on these two things, what you are trying to say is, okay, I will get a spectra, and the spectra will fall into either of these categories. Now why I'm saying is, what is the intensity of X-ray? Because when the X-ray goes and hits a particular atom or a molecule, what happens is it actually goes into its electronic environment. So the electrons rotating there, the X-ray goes and hits there, and depending on the density of this electron cloud over this particular atom, the angle of diffractions or the intensity of diffractions will be determined. Now based on this, so what we can see is, if we are getting a more you can say intensity of X-ray after passing through your sample at a particular spot, that means the electron density from where the diffraction has happened is very less, right, compared to another spot where the diffraction has happened and the intensity of X-ray onto the detector is very less. That means the diffractions or the electron density which this particular X-ray has experienced is way more, and that is why you are experiencing an diffraction of the X-ray which is high, and the end intensity of the X-ray which is detected at the detector is very less. Okay, so this thing should be clear, that what we are looking for and why we are looking for, right? But even if you see this, we saw this structure. In this structure also, it is not exactly the same which you are seeing it like you cannot draw as a geometrical shape like this, but your actual protein is something like this. So when your structure becomes something like this, then there are a lot of angles, internal angles also come into play, which result into different angles of diffractions, which result into different intensity of the X-ray passing through them, right? Now a brief outlook about why these structures or this protein structures are so difficult. So primarily you can see a linear chain. This is like a long chain of amino acid sequences one after another, and you can have as many as you possible depending on your or the protein you are looking for. But this same linear chain becomes folded by the virtue of different hydrogen bonds, and you can see they are no more linear, that they are becoming more like helical structures. And this secondary structure gets further, they get folded, and you can see this, and finally it will be into this quaternary complex structures, right? So when we are talking of this particular protein, as you can see here, we have to understand that this protein orientation, again for the same type of protein, the orientation will not be same. So what we have to do is, if you are trying to suppose identify a structure which is fast moving, right, so they are in free form, they can move in clockwise, anti-clockwise, or in different spatial directions. What will happen is, when you pass the X-rays through your sample, every time, due to the change in the sample orientation, the angle of diffractions and everything will keep changing. So the resultant informations, you will be confused, what is happening with my sample. Now on the contrary, what we can do is, if we suppose fix this structure into this orientation, so how do we do it? We actually form crystals. So when your protein is pure, what you do is you change the temperature and the crystallization is happening. So when you are making it as a crystal, what you know is this particular orientation is fixed. Now when I have a fixed structure, it is easy for me to identify or study its structural orientations, right? and that is why the crystallography, or you say the word crystallography, because you have to make your samples in the form of a crystal, and once it is in the form of a crystal, you pass your x-rays, or you put the sample in the x-ray crystallography, and what happens is your x-ray beams goes and then your structural elucidation starts. So this process of doing it starts with crystallization of your sample, right? Now when I'm talking of a crystal, now this crystal is basically a solid material, right? It's a solid structure, but when I'm talking at an entire protein, it's mixed of all the atoms and molecules of that protein. What we have to do is we have to narrow it down. So just imagine in your institute there may be, suppose, around 50 classrooms, right? And out of these 50 classrooms together pulled up a building, and which may be your entire campus, just for example, right? So if you are thinking that your entire institute building comprises of 50 units, so narrow it down to just one classroom where you are sitting right now. So one classroom is basically one unit cell which actually can be studied individually, or which can be focused individually, and the same thing can be repeated for all the 50 classrooms one by one. So if you can imagine this part, what we'll do is we'll take this one unit cell or one unit part of the lattice in the crystal, and then we'll understand the structure of the lattice, right? So once we know the structure of the lattice, it is much easier for us to get or to decipher information from one single unit, and then keep on pulling the data from other units and then get the entire structural elucidation done, right? So is an important, yes, obviously it's important for materials, and it's basically in crystal the advantage you have is you have a repeating unit. So when I'm talking of one unit as your classroom, you have multiple classrooms, so it's basically a repeat of this unit cell where you are sitting right now, right? And these repetitions will also give diffractions in the X-ray, right? And these repetitions will also give informations based on the unit cell. Now if you understand this unit cell, we can also understand why my data is getting repeated, why my X-ray is getting diffracted in the similar pattern in all other unit cells, and that will give us the final you can say the information which will be more like a repetitions in crystals, and this entire thing is called crystallography. Okay, so basically it is a repeating type of doing it, because we are not going into the holistic thing here. We are trying to get your sample into the form of a crystal, and once it is in crystal, it is in a solid fixed state, there is no movement, it is more or less in a fixed orientation, and then in this fixed orientation crystal we are trying to reduce it to one small unit cells or lattices, and then once we understand this lattice's behavior, it can be repetitive in the entire crystal, right? Now there is another way, problem with all this thing is rotational symmetry. So rotational symmetry is your object may rotate, or you can rotate it to see the different directions. Like if the same box, if I keep on rotating, what will happen? I'll check the symmetry, whether this particular box is symmetrical in all the directions or not. So if I keep on rotating and I check the symmetry, then also it will give me an information, right? And this information will be helping us to give in how many directions this symmetry is distributed. So if I keep on changing this, you see here I am distributing, so one big square I am equally distributing into small 4 squares, right? And then I'm again distributing this into triangles. So this is my distributions I'm making into finer or smaller units. And similarly for different structures, I can do this to smaller units, because the smaller units will give me an ease of understanding, right? So the idea is, if you know the lattice, you can minimize it to a smaller unit of the lattice, and then once you know the smaller unit, you know the structural orientation of it very easily, and then the same thing can be reciprocated in the other units. Okay, Now there can be also like rotations, not only in the angular but also it can be in one dimension. Like in one array also you can have certain rotations which comes in the form of a periodicity. So it is like at a regular interval you have these rotations in your sample. Now let's try to get a very schematic or a very simple drawing of the translations, right? How you are translating this lattice or this grid thing, and how you can see it in your sample. So first we have to understand what I'm talking of as a lattice. Okay, so you can see this is a grid. Basically you can see there are many lines drawn, and each of this is basically similar. You can see they're all of the same. So if you take them as small rectangulars, they're more or less of the same size. Not more or less, exactly of the same size. So if you place one, or if you know one, so I'm just taking out one point or each block, and if I understand this particular block, I can repeat it with other blocks, right? Okay, so I have to understand or start with one small unit, and then I can repeat it with other blocks, and I will get an array points, which is basically a lattice. So this is basically a lattice. So it was first a single spot, a single junction or a molecule I studied, and on this I went to the entire array of these junctions, or basically for this take as points, and once I complete this array of points, it will give me the lattice structure. Now this lattice can have different symmetry. It can be in the form of a square. As you can see, all the directions they are similar, so it can be in the form of a square. It can be in the form of a hexagon. As you can see, it is a hexagonal structure, right? And this is hexagonal. Why? Because we are seeing only one flat side of this lattice, but you also have a center, so it is basically a hexagonal with six-fold symmetry, right? So a six-fold symmetry will have this, and then we are narrowing it down to the smallest possible. So once we understand what is the lattice orientation, try to minimize it. So I started with this. I had this lattice array, and from this lattice array I am minimizing it to just 4 dots or 4 points in the lattice. So once I have minimized it, it is much easy for me to study it. So it is always difficult to study the complex structure, but if I try to bring it into a small units, as I was saying, so here if I start with, suppose I have to complete, or I have to teach in all the classrooms. So there are 50 classrooms, I have to teach in all the classrooms. So why not I start with one classroom, I dedicatedly finish for one classroom, then I repeat the same process in other classrooms, and eventually I'll complete it, right? So that in taking that similar idea, if I have a small lattice, it is easy for me to manage. It is easy for me to study how the X-ray diffraction is behaving once passing on to this lattice plane, and then I can do the same repetitions for other planes also. Then I can understand what the entire behavior of my crystal would be, right? So this unit cell is very important. Now what we have to also understand is the basic unit cell, it will have a lot of vectors. So if you talk of a small cube or a unit cube, it will have basically 3 vectors. Usually it is said H, L and K. So it is height, width and length, right? So height, length and breadth or width, whatever you want to say. So the same thing is annotated here as A, B and T. It is just a form of expression. You can people also say as L, K, that way also you can do that. Okay, H, L and K, that is annotations are also preferred in some of the textbooks. So it is up to you how you want to annotate. But what you have to understand is there are 3 vectors in 3 directions which are working here, and then you have to understand what are their orientations, because vectors will give the orientation of this unit cell which you are studying. So you see here, this is the 4 dots and the 3 vectors which I have drawn here. So 1, 2 and this is the 3rd vector in this direction. But if I make a cube, then my vectors are very clear. So you see here, 1 vector in this arrow, 1 vector so B is in this side and the C is in the upside. So this gives me the 3D information. So the idea is Why I started with dots? Because I wanted to give you an understanding of a plane, and once we have multiple planes we can join these dots to come up with a 3D structure. And that is how from single plane, if we understand the lattice orientation, we can decipher the informations and then interpolate it in the 3D structure, which is actually the structure present in your crystal of interest, right? Now, in this crystal lattice or in this small unit cell, you also have, apart from three vectors, you also have angles. So these angles are also not fixed. So basically you have three angles, alpha, beta and gamma, and this angle changes with different. Now we have to also understand what are these angles, right? Not only the vector direction but also these angles, because it will not be a fixed 90 degree or not be a fixed cube. It may be hexagonal, it may have different varying angles in different samples. So this angle identification understanding is equally important. Now just to give you an so whatever information we had, so this is like one plane, and you can have multiple of other planes also. By connecting different dots, you see different orientations and angles have come up. So this angles, because why this is important, it can be flat rectangular like this, or it can be something like this with a different angle, because your beta sheets, alpha helix will not be in the fixed orientation. You have to understand that. So if to understand their information, to understand their orientation rather, which is actually present in the crystal, what you have to do is you have to understand this angular part also. So a unit cell we have picked up, it is simple, it is the smallest unit possible. If it is a right angle, then it is very good, it becomes much more easy and it is simple. And the symmetry of the cell shape is consistent, that is in the best criteria. But at times this is not the actual story when you are using the sample, so they may change. But yes, if you keep that in mind, you can understand what type of your crystal system like. So coming back, so I think this crystal system in your class 11 12 you must have read what are different types of space lattices and crystal systems are there. So based on that, if just a quick recall, you can have a cubic, hexagonal, tetragonal, trigonal, okay, you can have orthorhombic, you can have monoclinical and monoclinic and triclinic. Now how they are classified, you can see depending on their space lattices and depending on the angles between them, and that is why I was emphasizing on the angle which you are trying to get in your unit cell. So you see, depending on the angle, the entire crystal system is changing, right? Obviously the body centered or face centered, what type of orientation is there in the space lattice, that is equally important, but again the angle cannot be left out as it is. So this is some of the example. These examples will help you, you can today you can go back home and you can take out the crystal structures of all this, and you can see how they are different from one another, right? And once you understand this basic crystal designing and the orientation, then it will give you a very so you have to be a little bit imaginative in this part, that in a 3D, in your mind you have to create a 3D, and in that 3D space how your protein is basically oriented. So this small informations will give you a better understanding. Now coming to the actual system schematic diagram. So this is the schematic diagram of the X-ray crystallography system, where your X-ray is generated. You basically have two focusing mirrors, one in the X and basically in the Y. Why it is X and Y? Because you have to scan your entire crystal, it is not a dot, right? So this X and Y mirrors, they will keep focusing and changing and different, so it will basically scan on the crystal which you are positioning here inside. And in this crystal what you have is you have the X-ray beam, and opposite to the X-ray beam you have the detector, right? So they work hand in hand, and you see here this is a change in angle. So either you change the angle in this orientation or you change the angle in this orientation, so that the crystal's angular change is there. and you study based, as you change the orientation, you study the pattern of X-ray diffractions which you are getting it. So there are two, as I said in the beginning, pattern means two things. What is the diffraction pattern, and one will be the intensity of the spot. So as you can see here, there are different spots of X-ray after getting diffracted from this crystal, and you see some are bold and some are small. So the intensity depends on the electronic cloud which this particular X-ray beam is experiencing. If it is less, then you have a bolder dot. If the electronic cloud is more, then you have a smaller dot of X-ray onto the detector, right? So I'll just show you a small video where you can see the entire workflow, how the X-ray is done. So if you can see my video, this is basically the sample where you are loading it, and this X-ray beam is coming onto your sample, and this is the detector, and they keep on changing. So as you see, the angle is being changed, and with change in angle you capture in the spectrometer the intensity, and you see this intensity are changing. So you see this is your signals. These signals are very broad, very high, and there will be certain noise also, and we have to understand this pattern based on the 2 theta. Now what is 2 theta? That is the angle of diffractions. I will come to it in a while, but this is how the entire system looks like. Okay, so this is basically depending on the the diffraction angle. Now in X-ray crystallography, if you see, this is basically the main idea where and how your diffractions are being taken care of, right? So just we understood about the atomic planes, or basically the crystal planes, or the lattice planes to be very precise. So consider this as lattice plane 1, consider this as lattice plane 2, and you can say the difference between these two will be atomic. So atomic planes also, because the lattice are all dots, if you remember. So within these dots, if you're connecting to different planes, then what is happening, your X-rays are coming and hitting onto different planes, and once it hits onto different planes, it is interacting with the molecules there, and in this interaction, what is happening, it is getting diffracted. So you see a diffraction taking place in plane 1, and again a diffraction taking place in plane 2. Now this diffractions, what we are more worried about, this theta. So what is the angle? So this is basically the angle of incidence, this theta, and this one is the angle of diffractions, and this angle of diffractions is usually considered as 2 theta. And why? Because if you see, this is the wavelength and this is the diffractions, and this is for the first on the surface, but as we go deep inside multiple layers or multiple atomic planes, you see this is the distance between the atoms. Here what is happening, the X-rays are coming here, but this portion, as you see, is not being traveled here in the first case. In the second one, you see this region is an extra distance which the X-ray is traveling, just because it has gone inside. So what is happening is, you have to see what is this lambda by 2, or what is the length of it, and depending on this, your diffracted X-rays will be generated. Now this diffracted X-rays, multiple of them are being generated, and they can form either a constructive interference or they can form a destructive interference, depending on their interactions with different atomic planes, right? So if you see, this is the X-ray generator, beam generator, this is basically your protein, and it is getting diffracted, and once it is diffracted, getting the spots, and the spots are again dependent on the constructed or the destructive interference which you are taking care of. Now XRD in grossly or in generally is considered as a non-destructive analytical technique. Means you crystallize your sample, it is a protein, right, in this case, and once you crystallize your sample, you pass on the X-rays. This X-ray is not going to damage your sample. Okay, so you get the data and you are not destructing or you are not damaging your sample of interest, and that is why this is considered as a non-destructive analytical technique, which is used to identify the crystalline material, right? Now if you see, this is basically one type which I have taken as BCC, that is body centered cube. Why this is body centered cube? You see the center, there is one atom here and it is linked with this, so it is called body centered cube. So this is one type of pattern you can see here. This is another face centered cube. In the face centered cube, the first one, it is in the center, but in the face centered, you see on the faces, so this is one face, this is another face, this is another face, so there are different face are there, or you can say the sides of the cube. On the sides of the cube, in the center, if you have any you say atom, then it becomes a face centered cube. And then this is again a hexagonal, you can see 6 are there, and in the hexagonal close packed, you see in the center there is another more, so you linked it, and this type of structure. So there are several types, as I showed in the previous table. This can be your, like you can go back today, and a food for thought is how this crystallization, how these lattice patterns are designed, and what is their internal architecture like. And crystals, why they're important? Because once upon forming a crystal, their patterns are very, very symmetrical, means their repetitions are as identical. So that is why if anything is in a repetitive structure, the complex structure can be narrowed down to a unit structure, that is a unit cell. And this unit cell will actually, if you can keep on repeating the study on this unit cell, you complete the entire structure. And that is the beauty of a crystal, which is not possible with amorphous or any other type of things, right? So that is why crystallography and forming of crystal is so very crucial, because it will give you not only a fixed orientation, but also it will give you the orientation which is repetitive in manner, and you can narrow it down or break it into a smaller unit cell, right? And also if you see of a polyethylene, so this is a orthorhombic structure, and you see here even this one can be also narrowed down to one small unit lattice structure. And this lattice structure, if you see, it can be repeated for the others also. Okay, now coming to the schematic diagram. So this is basically the X-ray, and this X-ray is coming to a particular atom, and in this atom you have the electronic cloud revolving around it. So this electronic cloud revolving around it will experience the incident X-rays, and this incident X-rays, once interacting with this electronic cloud, it will absorb it. It will absorb the X-ray, but at the same time this will become very unstable. So what will happen is, being unstable, it will again try to emit the X-ray out of it and become stable. But in the process, what will happen, due to this particular electronic cloud orientation, what will happen, the wavelength which is emitted out, or basically the X-rays which are emitted out, they will be in a diffracted format. They will not be the exact wavelength with which they have come and fallen onto this particular atom. And this change in wavelength and how they interact with each other will give you, or the entire, you can say, the information hidden of the structural orientations is hidden by the pattern how these wavelengths are emitted out from the atom, right? So if you see, this is the X-ray falling, and this is the angle. So coming to the more angular part of it, these are the lattice planes or atomic planes, which I have already discussed. So why I'm actually repeating it this time, because this is something which is the heart of X-ray crystallography. If you understand how this works, I think rest of the things will be very easy for all of you to understand. Okay, now there is D is the distance between two atoms or planes, and this is the angle theta. So this angle theta is the angle of diffraction, and this angle is the angle of incidence, because X-rays are coming from this side, hitting onto this lattice plane and then getting diffracted. And on the first plane, you see this is just hitting and going, so it's more like a reflection sort of. But here it is little more, because this distance it is traveling. You see the region within this dotted line, it is the wavelengths are traveling extra, and this extra traveling will force them to give that diffraction. So this is given by the d sine theta. So sine theta is this angle of diffraction, and d is the distance between the atomic planes. and from here what we get is we set multiple, so like as and when it goes deep inside, this angle as well as the distance keeps on changing, and this too will give you the expression which is called Bragg's law, which is depending on the lambda you can have this expression. So mainly it is dependent on what? It is dependent on this theta. So this theta is what is more important, and based on this theta, because d will be fixed, you know the distance between the interatomic distance, and the wavelength, this is the X-ray which you are using it, so that this all part will be fixed. And as I said, HKL, this is basically the three vectors or three dimensions of the lattice. Like as I showed is ABC, similarly it is HKL representation, and then you have the theta in all the three vector side, which can be pulled together to give you this expression, right? So just I think we will also continue in the next class, but before we end, there's a small video where I'll show you how the entire workflow works, and I think it will give you a very clear understanding how this is happening. So you have different types of, it may be crystals or it may be finer structures. So you can see here, this is basically all the atoms and they are in a crystal formation, right? So in crystal formation, you see the pattern, the patterns are all fixed, which I was trying to explain you. And this, each of this, you can see here the nucleus and the electron environment. So electrons revolving around them. And this electronic clouds, when you have the X-ray, so this is basically the lambda, so the one wavelength, and when the lambda passes, so again coming to this point, why X-ray is used? Because the distance between these two atom, or the distance d, is equal to the lambda of which is equivalent to the X-rays. So the distance or the wavelength and the distance between two atoms are more or less same, and that is why X-ray is able to go inside and give you this information. So as the X-ray beams are changing, you see the diffractions are happening, right? And these diffractions can be constructive, so you have multiple wavelengths falling constructive and you're getting a higher interference. You see, this is called a constructive interference. You have lot of wavelengths and they are constructive because the phases, the peaks they are matching with one another, and that is why the resultant amplitude is high. But what happens if this phase doesn't match? So they are in opposite phase, what will happen? They don't match, they form like this, and the resultant will be a destructive interference, right? So this is something very important. If you are getting a constructive interference, the phases are matching. If you are getting a destructive interference, so you have one phase like this, and you see the similar opposite phase is also there, they are nullifying to each other and you are getting a destructive interference there, right? So when you see the X-rays are coming, the electron is absorbing that energy, it is becoming very, very unstable, and usually what is happening, it will try to gain its stability by the process of emitting the X-rays. So these X-rays are again emitted out, and this electrons are becoming, so this process is called elastic scattering, right? This process is called elastic scattering. Now here, this is what is the crystal, so different crystals, and the distance between two is the d, which you can see here, this is the distance or interatomic distance. And when you have the X-ray passing through different planes of this lattice, you see there is a change. So these are the planes which I was showing. So first of all, if it is a constructive interface, you will get a brighter spot, and if it is a destructive interface, and this is basically the degree or the angle, you can see the angle of diffraction. So this is the 2 theta which I was trying to say. So 2 sine theta will give you the angle of diffraction here. And if you have a constructive interface, what is happening is here, once you are having a constructive interface or a constructive thing, then the diffracted phase or the diffracted wavelengths, X-rays, should have a same phase. Like you see here, these are diffracted X-rays, right? And you see the phase, they are matching exactly to each other, and that is why you are having a constructive interference, right? And to more to it, if you see the planes, if you actually want to know why the distance between them is increasing, so there is one straight line like this and this straight line like this is the lattice angle. The first one is coming, and you see this is actually getting in the form of a diffraction, but it looks more or less like a reflection, sorry. But here in the second case, in the second plane, you see this is half of the lambda which it is traveling extra, and similarly on the other side it is diffracted half of the lambda which it is traveling extra. So if a lambda, so together lambda half gives you one full lambda, and that is why you see this phase and this phase are in sync. But if suppose you imagine this and this doesn't add up to one single lambda, so this may be one lambda and this may be half lambda or 1.5 lambda, then the resultant diffraction wavelength will not be in the same phase, because this path length is not matching, or it's not exactly one lambda difference, and that is why it will not match with the same phase, and that is where your constructive and destructive interference comes into the picture, right? So this is something which you understand here, and this keeps happening for all the planes as we go. So if you see here for this plane, you see the lambda. So this is one complete lambda which it has traveled, and in this one complete lambda, so this is one full lambda, this is another full lambda. So this again is a constructive interface, because if you remove this to complete lambda, the phase here and the phase starting here is almost matching to the phase here and the phase starting here, right? So if this is not complete, then the constructive interference will not happen. So for each of these planes, so what is different is this lambda which it is extra traveling, if it is complete to one full lambda, and then a constructive interference is likely to happen. So okay, this thing I think is very important concept, and that is how you understand and how you calculate. This is the D, this is the lambda by 2, this is the diffraction wavelength, diffracted wavelength, and here if you see this is basically the theta. If you draw the planes on which the angle of incidence and diffraction has happened, so you can see these three things are happening, and together if we try to see with the lattice dots, you get the expression as sine theta lambda by 2d. And if we simply try to move it to with respect to the lambda, you have your 2d sine theta, and this is given by Bragg's. So Bragg's basically this is father and son, sorry for this. So Bragg's, this is father and son. Sir William Henry Bragg and Sir William Lawrence Bragg, they together came up with this diffraction, and it's called Bragg's law, right? And for that they received Nobel Prize. So I think with this I will stop today's class, and will continue from here onwards in the next class.